Hello, and welcome to Spygen's training modules. We are pleased to have you here to learn more about Spygen's environmental DNA technologies and protocols. This training has been designed to guide you from the foundations of eDNA to its practical applications in the field. So, let's get started. In this module, you'll discover what environmental DNA is, explore the key concepts behind this technology, and get a quick overview of the full journey of a sample, from the field through the laboratory, all the way to the final biodiversity report. First things first, DNA, or deoxyribonucleic acid, is the molecule of life. It's present in every living organism on Earth, from the tiniest bacteria to the largest whales. Think of DNA as a universal instruction manual. It contains the genetic information that makes each species unique and tells each organism how to function, grow, and reproduce. This genetic code is like a biological barcode, unique to each species that can be used to detect and identify it. And so, this brings us to environmental DNA, which is the DNA that can be extracted from environmental samples of marine water, freshwater, and even soil, without ever touching, seeing, or disturbing the species we are studying. Through eDNA analysis, we can identify the species present in a site simply by collecting the DNA traces they leave behind in their surroundings. This DNA comes from different sources, such as whole microorganisms, or biological traces left behind by larger organisms. When organisms swim, walk, feed, reproduce, or simply exist in the environment, they constantly release DNA into their surroundings. Imagine a fish swimming through the water. It continuously sheds scales, releases mucus, defecates, and breathes. All of these processes release DNA into the surrounding water. It's like leaving a biological signature everywhere they go. Now you can see why this technology is so valuable. By collecting and analyzing the DNA traces left in the environment, we can detect which species are present without ever seeing them, catching them, or disturbing their habitat. And Spygen's environmental DNA technologies are particularly powerful for detecting species that are rare, elusive, or cryptic, whether they're protected, threatened, or invasive. This approach enables low-cost, large-scale, and standardized biodiversity inventories, all without negatively impacting ecosystems. You can survey any type of aquatic ecosystems, from a small pond to a large marine area, by simply filtering water samples rather than deploying nets, traps, or underwater cameras for weeks. At Spygen, we have been at the forefront of biodiversity monitoring since the very beginning, pushing eDNA technology to its limits to deliver the most comprehensive biodiversity inventories to our clients and partners. So, when we filter water at a site, we're collecting DNA traces left behind by different species. But here's the key question: how long do these traces last in the environment? Studies show that in aquatic environments, DNA traces persist only for a few hours to several days, depending on various conditions. That's why eDNA is like a snapshot. It captures which species were present at that site and exact moment in time. eDNA provides a snapshot of recent biological presence, not a historical record. Once released into the environment, DNA begins to break down. Several factors influence how long eDNA persists, including enzymes, bacteria, UV radiation, temperature, and salinity all contribute to degrading the DNA molecules. All affect how long DNA remains detectable. Another key factor is that eDNA does not travel very far under most conditions. It tends to remain concentrated near the source organism, which is why we describe it as highly localized. That's why we collect water subsamples from across the entire water body, to provide a more complete and reliable snapshot of biodiversity at the site. By contrast, fixed-point sampling, for instance, can be misleading. If you only collect water from a single spot, you might miss species that are few meters away, and that can distort your conclusions. Let's now talk about two more key concepts: false negatives and false positives. A false negative happens when a species is present at a site but doesn't show up in your results. A false positive is the opposite. A species appears in your results but isn't actually there. At Spygen, we've developed our technology to minimize both. Still, it's important to understand why they occur and how to prevent them. First, let's look at false negatives, when a species is present at the site but not detected by the technology. These can happen for several reasons, including wrong primer choice. Each primer is designed to amplify specific taxonomic groups. This ensures we can detect the greatest number of species with the fewest false negatives possible. At Spygen, our primers are rigorously tested and proven. They represent the cutting edge of primer technology in the field. Poor sampling strategy. Sampling is a critical step in eDNA analysis, as it ensures that sufficient DNA is collected for the subsequent stages. If you collect water from a location where the species was not present, you risk missing it. For instance, relying solely on fixed-point sampling increases the likelihood of overlooking certain species. Biological factors. Some species naturally release less DNA. Reptiles, for example, shed much less than mammals or fish, making them inherently harder to detect. Gaps in the reference database. If a species isn't included in any database, its DNA sequence will appear as an unidentified sequence rather than a confirmed species detection. This is why reference databases are so critical for reliable results. Over the past 15 years, Spygen has built one of the largest proprietary, field-tested, and taxonomically verified databases in the industry, and we continue to expand it every year to improve detection coverage. As for false positives, when a species is detected even though it isn't truly there, it's important to understand that eDNA detects genetic material, not living organisms directly. There's a main situation that can lead to these detections: DNA from contamination sources. If proper field protocols are not followed, DNA from outside your study area can contaminate samples. For example, sampling equipment previously used at another site without proper cleaning. This is easily prevented by following proper field protocols, such as using clean, dedicated sampling equipment and applying proper handling procedures between sites. This training ensures you know exactly how to avoid these issues. Another factor to keep in mind is season. During reproduction, many species release more DNA through gametes and reproductive behaviors, making them easier to detect. For monitoring programs that track changes over time, sampling during the same season each year ensures differences reflect actual biodiversity changes rather than seasonal variation in DNA shedding. Our comprehensive approach helps reduce both false negatives and false positives. Good field practices are the foundation of reliable results. Follow sampling protocols precisely, maintain strict decontamination procedures, and use only single-use materials between sites. Store samples properly with the conservation buffer, and document all conditions thoroughly, including location, time, and weather. Whenever needed, our experts will support you in designing sampling protocols that minimize error risks for your specific project context. By understanding what can affect eDNA results, you can minimize risks. Now let's quickly follow the journey of a sample, from the field, through the lab, all the way to your biodiversity report. So, we know that sampling is one of the most critical stages in environmental DNA analysis. A well-designed sampling protocol significantly increases the amount of DNA collected from the environment, leading to more accurate results. That's why we help you define the best protocol for your project. You'll get it through the training provided in these modules, the resources on our website, and the direct support of our experts when needed. We send you all the sampling materials, and with this training, you'll see that collecting samples yourself is simple and straightforward. It's just filtering water through our filters and ensuring you minimize contamination. Once you've collected the samples, you'll add our conservation buffer to the filters. This can preserve the DNA for months at room temperature. Then, you send the filters back to us. In our labs, we begin by extracting DNA from your filters. This process isolates pure DNA by breaking open cells and washing away sediment, salts, and organic matter. Then we come to two important steps. First, amplification. To detect your target groups, the DNA has to be amplified. For this, we use high-performance primers, covering a wide range of species. You can check our website for the full list of biological groups and organisms we can detect. In this process, we run 12 PCR replicates. That means your DNA is amplified in 12 independent subsamples. This boosts detection rates. Remember, more replicates mean higher detection probability. Next, sequencing. Here, we read the amplified DNA. In other words, we decode the genetic information. Sequencing depth drives detection power, and the principle is simple. The more DNA strands you read, the greater your chances of detecting rare or low-abundance species. And at Spygen, we use a high sequencing depth 5 times higher than basic protocols, so we can detect more species, including the rarest ones. Finally, the sequences from the lab are matched against the reference database. This step connects each DNA code to the species name. We rely on a combination of publicly available reference databases and Spygen's proprietary database. But no reference database yet includes the genetic information of every species on Earth. If a species is missing, we can work with you to obtain a small tissue sample and add it to the database. A complete and well-curated database is essential for accurate results. In addition, our experts carefully verify that every identified species in your project is consistent with the environment you are studying. We reach the final report stage, where the biodiversity of your site is revealed. In this training, you'll also learn how to interpret the results we deliver to you. Be aware that not every DNA detection can be identified at the species level. Sometimes it's only possible to match a detection at a higher taxonomic level, such as genus or family, because a species may share the same sequence or reference data isn't sufficient for finer resolution. The good news is that these cases will become less frequent as reference databases grow, and at Spygen, we work every day to ensure you benefit from the most complete databases available. When you choose Spygen, you're not just getting eDNA analysis, you're getting 15 years of innovation, the industry's most comprehensive reference databases, expert validation at every step, and a proven track record of detecting the species that matter most. Stay tuned.